Good evening, everyone. This is Gayatri Agrawal, and today I'm taking a presentation about Bacillus thuringiensis. So, next slides. Bacillus thuringiensis. It is short form we can call as BT. It is ubiquitous spore-forming soil bacterium that is well known. It is basically it is insecticide, where it is an active form wide range of pest insects. It is used as pesticides also. The potential of the BT can be used in insecticides was recognized in early 20s. It's not now, but from the early 20s, it has been used in the agriculture. Based on the pesticides have been commercialized. The advanced molecular biology has made it as a tool which has many benefits, which can protect the agriculture from where the insects. It is used to kill the beetles and the larvae of the mosquitoes and black pearls, but also it is also used as biofertilizer. Next is the first BT crop was commercialized in 1995, but today BT corn and cotton, soybeans cultivated in 76 millions in 27 countries. Multiple factors drive the stretch of BT as the significant focus in the triad discovery. It is used as biofertilizer in crops with their implications in the control of biopathogens in the insects and pests. That is the main. What does BT does? BT is a species of the bacteria that lives in the soil. It makes the protein or toxic to some insects when eaten, but not others. The proteins are non-toxic to humans because, like mammals, we cannot activate them. The main thing is it eats the bacteria uh, which is in the soil and makes the protein as a toxic, which prevents the which is used as a biofertilizer. Where Bt is a gram-positive bacteria characterized by producing paraprozole, which insecticides activating during sporulation. Bt comes in a different variety. It is mainly used as sprays. Bioinsecticide in agriculture, forestry, mosquito control, where we can see nearby our homes also sometimes they come in a government only provides where we can control the mosquitoes. It is also in the spray form, it will come and the mosquitoes prevention also can be done by the BT. Intensive planting of the BT crops inevitably creates strong selection pressure on the target insecticides. There are the type of Bacillus BTS, it has been used in agriculture long time because of its insecticide protein, which is made valuable environmental friendly biosecticides, in which BT plants usually BT. The most common crops are corn and cotton. These are the main two crops where corn and cotton the most BT has been used. The mode BT is the three main conditions where maximize the effectiveness of the refuse strategy. Recessive in inheritance of the resistance phenotype, low frequency of the initial resistance alleles, and abundant susceptibility insects provided by an BT host plants nearby. Strains, there are the BT strains have been developed by the laboratory selections. Other insects such as Pileatal selector and cabbage, looper, and have evolved in the BT sprays in the open fields and greenhouses respectively. The applications of the it is widely used as bioinsecticides to control the infestation of the butterfly caterpillars also. Caterpillars eat the spores sprayed on the plants and get killed. BT produces an insecticidal protein during sporulation. When the sporulation happens, the BT produces an protein which helps the caterpillars not to kill the leaf. The application of the biotechnology in agriculture involves scientific techniques such as genetically modified organisms. The cotton pest, it helps in the modifying plants, animals, and microorganisms and improve their agriculture productivity. The role in the the main BT, the role is synthesis of different insecticidal proteins named CRI, VIP, CYT. They are able to kill the different insects in order to. These proteins have been extensively used in the control practice in agriculture sprays expressed in the genetical modification. The era of the BT had been beginning in the 1901. The Japanese scientist Mr. Shengtang Ishwat isolated the bacterium from dead silkworm larvae. So, last slide, sir. Insect cadavers. The search of the bee. No, sir, last. Insect cadavers, I mean, sir. No, sir. One more. The sixth slide, I guess.
Sixth.
So, sixth slide, sir. Okay, the BT, the insect focus on the host rather than the habitat, because BT cause the death of the host insects in 3 to 7 days. One of the most rewarding easy source of the BT in the insect cadavers. Thus, reportedly, the BT could be more readily isolated from the insect cadavers rather than from the soils. BT next slide. Next slide. The next slide. The strains are isolated from the naturally occurring soil bacteria that are used in the insect pecks, agriculture crops, stored fruits, ornamental plants, and the water bodies around the homes. Where we have small the differential strains have specific toxicity, particularly types of insects depending on the specific protein that are produced. BT is considered as almost ideal agent for the pest management because of its combination insecticide specific, lack of toxicity to the humans. Next slide. Next slide, sir. Here you can see the BT is this thing. It is a gram-positive bacteria where we have the ribosome, cell membrane, cell wall, DNA, and endospores. BT is considered as a safe for the humans. On the leaf, you can see where the insects are biting the leaf, but when the spray of the BT has been used, these insects will be killed from the insects will be killed, and the leaf will be alive, like without any damage to the leaf. Thank you, sir.